Great. Now we'll move into the research talks. So it's my pleasure to introduce the first of our research talks, Dr. Shamima Rahman, who is a professor of pediatric metabolic medicine at the University of College of London. So Dr. Shamima, take it away.
Thank you very much. Thank you to Greg for that really inspirational talk. It's a very hard act to follow. And thank you to Casey and Sophia for the invitation to speak to you today. So I've been tasked with talking to you about some of the diagnostic resources for Leigh syndrome that we've been developing over the years with a very large community of global partners. And it's very exciting to be able to talk to you during World Mitochondrial Disease Awareness Week. And the picture in the middle there is some of the UCL team when we lit up the portico this time last year, and we're going to do it again tomorrow. These are some of our disclosures. To go into an overview of the talk, as I'm the first person speaking in this research session, I thought I would take the liberty of thinking about some historical perspectives on Leigh syndrome, and particularly the clinical, biochemical, and genetic complexity that leads us to need these diagnostic resources, and the tools that we've made, which include an international classification, the Leigh map, and also gene curations of more than 100 disease genes. So who was Leigh? Dennis Leigh was a British neuropathologist working in the 50s and later, and in 1951, he reported the pathology of a 7-month-old baby who'd very sadly died after a 6-week illness with sleepiness, not feeding well, poor vision, and also deafness and limb spasticity. And he reported a very distinctive neuropathology, and on the right of this slide, you can see some of his own hand-drawn illustrations. What happened in the subsequent years is that about 20 years later, someone noticed that the appearances of the brain were similar to what we see in thiamine deficiency, which can occur as something called Wernicke syndrome in alcoholics who are nutritionally deficient. But going on into the 70s, it became apparent that there was evidence of mitochondrial dysfunction in patients who had Leigh syndrome, including ragged red fibers in their muscle biopsies, complex 4 deficiency in one patient in the Netherlands, and pyruvate dehydrogenase deficiency in another patient. And then also other biochemical defects reported, including biotinidase deficiency and mitochondrial complex 1 deficiency. So this was a very large number of biochemical defects already, which was causing a lot of confusion. And then in 1991, so 10 years after the first human genome mapping project had been completed, that was the mitochondrial genome, mutations in the mitochondrial DNA were linked to Leigh syndrome. So this is when I came into the field in the early 1990s. And at that time, we had a limited number of causes of Leigh syndrome, and we were able to show that 10% of the children who had Leigh syndrome in Australia, where I spent a year, had mutations in the MT-ATP6 gene, a subunit of complex 5. It's taken far longer than we could ever have imagined to try and understand only, I think, a little bit over half of the causes of Leigh syndrome. But during the 1990s, 2000s, and the last couple of decades, we're able to see that there is quite a characteristic clinical presentation in a lot of affected children and adults. And one of the big breakthroughs was the advent of first computerized tomography, CT scanning, and then later magnetic resonance imaging, MRI brain scans. And these are some MRI scans where there are bilateral, symmetrical, hyperintense lesions in the basal ganglia and in the brainstem and other parts of the brain, which can help to make a diagnosis in life. So we weren't needing to wait until after death to know that it had been Leigh syndrome. And that was really important in terms of trying to develop treatments, obviously. Also, in that period, it became obvious that there are some areas of higher prevalence of Leigh syndrome, such as in the Saguenay-Lac-Saint-Jean region of Canada and in the Faroe Islands, but 2 studies from very different parts of the world in Sweden and in Southeast Australia had shown that the minimum birth prevalence is something between 1 in 30 and 1 in 40,000. So altogether, Leigh syndrome is not that rare, but individually it is lots of different rare diseases. And one of the intriguing things is that in many, many cohorts across the world, it seems to be there is a slight excess of affected boys for reasons that we don't understand. And it can't really be explained by X-linked genes. So we became increasingly aware that we couldn't find the genetic causes for a lot of cases in the mitochondrial genome, and so the nuclear genome must be involved. And over the years, Vamsi Mutha has done amazing work showing that about 1,500 nuclear genes are needed for mitochondrial function. So that's 7% of our nuclear genome is dedicated to making a mitochondrion that works well. And we think about 1/3 of Leigh syndrome is caused by mitochondrial DNA pathogenic variants, and the remaining 2/3 by nuclear gene variants. So if we go along back to that 70-year history of Leigh syndrome, the first nuclear gene defect was found in a subunit of complex 2 in the SDHA gene by the Parisian group led by Arnold Munich and Agnes Rotig. And that was interesting because that's the only subunit enzyme of the respiratory chain that doesn't have any mitochondrial encoded subunits. And then what really changed things was towards the end of the 2000s, and certainly from 2010, where something called next generation sequencing came along, and that allowed a period of very rapid gene discovery. So this uses new sequencing chemistry so that you no longer had to sequence 1 or only a handful of genes at a time. And so you could do dozens or hundreds of genes in a panel, or you could do the whole exome, which is the coding parts of all 20,000 genes in the genome, or the whole genome, which is just everything in the genome. And this really was a diagnostic revolution, and it does mean, say, for example, in the UK now, we can order a genome sequence for a child and their parents for any child who's very sick and get a result within a few weeks. And it's also meant that we've moved away from muscle biopsies as a diagnostic tool. So what that meant was from that initial picture I'd shown you from the early 90s to now, if we look at the mitochondrial gene variants in the pink and the nuclear gene variants in green, and we also know that there is a continuum of what Leigh would have described as tight Leigh syndrome and neuropathology, and then something that I coined years ago as an artificial term called Leigh-like syndrome, which I'm really trying to remove from our vocabulary nowadays, and going right out to other mitochondrial brain diseases. We look at all of this continuum, and the same genes can cause Leigh syndrome or other mitochondrial brain diseases. Then we can have complex 1 deficiencies, 2, 3, 4, 5, multiple respiratory chain deficiencies, either because of problems with maintaining the mitochondrial DNA or with its expression, so translation and synthesis of mitochondrially encoded proteins, problems with the mitochondrial membrane lipids, cofactors, vitamins, pyruvate dehydrogenase, and with some miscellaneous other gene mechanisms that I don't have time to go into today. So that means that we're at a count of more than 100 genes causing this one disease. And that does lead to quite difficult diagnostic problems, because if we sequence the whole genome and find different variants, how can we be sure that the variants we found are the cause of the disease in the child who is in front of us? So we've been working in more recent years to ensure that all of the information I presented to you so far is used to expedite genetic diagnosis for patients. And this in turn will help recruitment into natural history studies and clinical trials. And you will have seen from Dr. Gray's presentation how important natural history studies can be to facilitate the clinical trials themselves. So I'm going to show you some of these diagnostic resources that we've been developing. So I've shown you a large number of the different ways that Leigh syndrome can happen. This is a cartoon of the mitochondrion, and problems can be in the respiratory chain OXPHOS enzymes themselves. So it could be energy deficiency, but also in other parts of the mitochondrion. So this could be about oxidative stress, or in some cases, and I know there's been a chat about ECHS1 deficiency, it may be because of accumulation of toxic metabolites. And from the work of Vamsi Mutha, there's some evidence that maybe it's direct oxygen toxicity in the context of a genetic cause of Leigh syndrome. So an important issue was to classify all of these diseases. So we took the broader group of all of metabolic disease, about 1,500 in total, and divided them into 24 groups, 6 of which are mitochondrial, I'll show you in a moment. And it's a continuous real-time revision and update, and we're adding new genes at about 60 a year. And this is the URL. We've made it freely available, and anyone can go and have a look at it. The Sunburst chart is interactive. You can click on it and pull up a table that lists all of the genes in a particular piece of the pie of that chart. So the mitochondrial diseases within the ICMD include mitochondrial DNA-related disorders, nuclear-encoded disorders of oxidative phosphorylation, disorders of mitochondrial clove-factor biosynthesis, of mitochondrial DNA maintenance, replication, and gene expression, and then some others that we've lumped together, really because otherwise we'd have had a huge fragmentation of different causes. And why does this matter? Well, we are working with providers of terminology for electronic patient records so that these terms can be used as diagnostic codes for patients, because that's going to be really important to be able to go back and retrieve patients with particular diagnoses who are eligible for particular natural history studies or clinical trials, because at the moment it can be quite clumsy and clunky to find the patients and to contact you. And I'm sure you'd like to be informed as soon as possible. So that's a piece of work that's going on based on the ICIMD. So we've talked about the clinical complexity of Leigh syndrome. Although there are some characteristic neurological features, we've heard from Greg's talk that the initial symptoms can be very non-specific, and there can be diagnostic delays and odysseys, which hopefully are being speeded up by the access to next generation sequencing that is becoming available in many centers in the world. But we do know that in addition to the neurological problems, there can be problems in other parts of the body, such as the heart, kidneys, diabetes, anemia. And really, it's been said that the mitochondrial disease can present with any symptom in any combination, affecting any organ or combination of organs at any age, and that also applies to Leigh syndrome. I thought about turning this on its head and asking, could we actually use this complexity to try and expedite diagnosis for patients? So I'd like to introduce you to a concept of phenomics. And here I was just manually mapping out genes that were causing mitochondrial disease as they were discovered and linking them to particular organs that were affected most in those particular genes. And this was a manual exercise and obviously not very efficient. But phenomics is the systematic study of all measurable physical and biochemical characteristics of an organism or a person on a genome-wide scale. So we then went on to create something called LeigMap, which is a computational diagnostic resource. And the question we were asking was, is it possible to harness the phenotypic information from affected patients to enhance the diagnosis? And this would be a tool to be used alongside next generation sequencing, not instead of. And so we did a systematic literature review initially of 1,000 papers in the literature, but eventually for all of the disease genes for mitochondrial disease up to 3,000 papers. And we needed a common language. So we used this thing called the human phenotype ontology that you can see at the bottom of the screen here, or HPO, and mined the data and then mapped it onto this existing map called RECON, which is reconstitution of human metabolism, now up to RECON 4IND. And we used the virtual metabolic human in collaboration with Professor Ines Tilia, who's now in Galway. So I just want to show you how LeigMap works. So we've made a model of the cell, and you can navigate like you can through Google Maps through the cell to find different parts of the cell and go down to the mitochondrion, find different enzyme complexes, and keep navigating to more and more detail, and you can pull up a specific gene that might be that you might find variants in a patient, and then that pulls up this submap on the left here for that gene with lots of external links. And then you can scroll down to the bottom of this. It's also got lots of information, mode of inheritance, clinical features, MRI appearances in patients. But at the very bottom, we've got the associated submodel. And if you click on that, that pulls up every phenotype ever observed in patients with defects in that particular gene. And that can be used to help match whether you've got the right gene variants for the patient that you're trying to find a diagnosis for. So I move on from navigating the Leigh map. You can also query it. So here you can put in search terms. So they're being typed into the blue box on the left. So you can put in a gene such as SDHA, and that will take you to SDHA in the map. And then you can do exactly things that I showed you in the previous video. And then you can also query phenotypes that an affected patient may have, and then search for those, and it pulls up genes with each of those phenotypes. In the interest of time, I'll move on, but you can see there's the URL there, and again, we've made it freely available. We're trying to make everything that we do freely available to the public. So we also tested the Leigh map, and 2 people who were not even clinically qualified tested 20 anonymized case histories where they had the history and some biochemical information, but nothing else. And they queried Leigh map, and they were able to find the right gene in the top 10 in 80%, and that was better than any of the doctors I know, myself included. So that's the Leigh map, and again, it's free to use if anyone wants to use it, but it is not a replacement for seeing a doctor or for having genetic sequencing done. So the last thing I want to tell you about is gene curation. So with Marnie Falk at CHOP, Joe Guy in California, in Los Angeles, we formed a huge consortium of people, the Mitochondrial Diseases Gene Curation Expert Panel within the ClinGen environment. I've shown you some of our biocurators here and where the people are in the world. And so coordinating calls with people in San Diego all the way through to Melbourne can be quite challenging, as you can imagine. What we aim to do was to evaluate the evidence supporting the relationship of the more than 100 genes associated with Leigh syndrome to Leigh syndrome using a very robust systematic framework called the ClinGen Gene Clinical Validity Curation Framework. And we had all of these expert volunteers. We agreed a definition of Leigh syndrome. The biocurators curated the genes, then presented them at monthly curation meetings. The Mito GSEP voted on the curations, and then we published these again freely available on the ClinGen website. The first thing we did was to redefine Leigh syndrome as a spectrum because of the reasons I showed you in that earlier cartoon. And we've removed the Leigh-like terminology, and we just have the Leigh syndrome spectrum of all diseases that would fit in with this definition that I've got on the right, and it's in our recent paper in Annals of Neurology, so I'm not going to read it out to you now. And this is another cartoon just showing you more detail of the curation process. And to go through 113 genes, it took us 4 years. This is the way we did it. We developed a matrix for assessing evidence at a genetic level for the nuclear genes, another one for assessing genetic level evidence for mitochondrial genes, and for each of the genetic evidences, the maximum was 12. And then we also developed a matrix for the experimental evidence associated with each of those genes with a maximum of 6. So the total maximum was 18 possible. And so from 12 upwards, we would call it a definitive association. Between 11 and 17 would be a moderate association. Between 7 and 11 would be moderate association, and below 7 would be limited association. And these are all 113 genes that we assessed, drawn out on that bar chart. And basically about a third of the genes we assessed had a definitive association, a third a moderate association, and a third a limited association. And that work has recently been published in Annals of Neurology. And if anyone can't access it, I think it's freely accessible to anyone actually in PubMed or in Google Scholar, so you shouldn't have any trouble getting hold of that. So I just want to conclude by saying Leigh syndrome, I believe, is a paradigm of primary mitochondrial disease, and it's the most common clinical presentation, sadly, in children, particularly in the clinic that I work in, that there is all of this heterogeneity and complexity at clinical, biochemical, and genetic levels. We have more than 100 disease genes, which makes things even more difficult. I've shown you 3 diagnostic resources, ICIMD, LeMap, and ClinGen. We've curated 113 genes for Leigh syndrome and have now moved on to the whole of mitochondrial disease and committed to curating another 250 genes. I believe that this work, which is done by a huge number of people, will facilitate diagnosis of future cases and inform gene panel design, and also, importantly, inform eligibility criteria for natural history studies and clinical trials. I'd like to thank the enormous numbers of people who worked with me on this project, and the people who funded the ClinGen projects, and also the LeMap work and the ICIMD. And thank all of the children and their parents and families affected by Leigh syndrome that I've met over the last 30 years, and all of you for listening. And I know we're going to have a question session a bit later on. Thank you very much.